Okay, in this lecture, we're going to learn about the brain's chemical senses, so our sense of smell and our sense of taste. And then I'm also going to make a video about our cognitive skill of attention. So our chemical senses can detect different parts of our environment than our vision and our hearing. So you can see all these different things that should bring up strong memories of smells in your mind. And you could probably think about the taste of some of these things as well. And we're going to cover today how the smell and taste systems of the brain accomplish these goals. So our chemical senses' job is to monitor the environment for the chemical contents, and we want to, in general, evaluate our potential foods. So we want to taste things, and we want to, based off the taste, know if we should be eating them or not. And we might even smell them beforehand. So you can see how this would be an adaptive process because you don't want to eat something that could be poisonous. Whereas smell responds to chemicals that are embedded in the air, taste responds to the same kinds of chemicals but that are diluted in your saliva in your mouth. And so we'll see how these two different types of receptors in the nose and the tongue can do these two different jobs. You can see here the general anatomy of where these receptors are. So here are the primary receptors for olfaction, and here are the primary sensory receptors for gustation. When we experience something that we like, when we go to a restaurant and we eat a delicious pizza, we're getting this multimodal impact on our brain, and we need to consider a bunch of different aspects of that in addition to only the chemicals in the taste and the smell. So for instance, the appearance and our current amount of hunger and also our attention and preferences can influence how we perceive the food to taste or the flavor of the food. Even the sound of the cooking food can have influences as well. And smell and taste interact in such a strong way that, in fact, without smells, we have a difficulty distinguishing different tastes. So you probably have all done an experiment in science class as kids where you could not tell apart the difference between apples and onions based only on their taste and texture if your nose was plugged. And so these different senses are working together to create this perception of flavor. So this is the kind of disconnect between sensation and perception, is that all these other aspects of your cognition can influence your perceived flavor of a food. As we know, when we start eating pizza and when we get to the 10th slice, it stops tasting quite as good. So first we'll talk about the olfactory system. We have this air passage that goes up through our nostrils, and it brings the air against this area in the upper nasal passage. And we have receptors that are dangling down into our nasal passage through a piece of bone. So you can see here's the piece of bone, the cribriform plate, which is part of our skull, and dangling through that bone are the axons of these cells, which are called olfactory receptor cells. And you can see these olfactory receptor cells have little cilia, little tails or hairs that stick out into the nasal passage and can pick up the chemicals that go by. We have about 350 different types of these olfactory receptor cells, and we're only starting to understand the differences, and rats have even more of them, so it's an even harder problem to solve. Once this information leaves these olfactory receptors, it synapses into the area here called the olfactory bulb, which is on the underside, the ventral surface of the brain. And from there, the neurons send axons into the brain itself, first to the amygdala, then to the thalamus, and then to the orbital frontal cortex. So smell is unique in that it doesn't directly go to the thalamus, but first stops off in the amygdala, which is part of the limbic system associated with our emotion and memories. And this is why sometimes smells can bring out these whole vivid memories, because of this very close connection with our limbic system. So we have these different types of olfactory receptors, and you can see that they pick up particular odors, and so different receptors will pick up different odors. But just like our cones in our eyes, each smell, like the smell of roses or the smell of rain after it rains, would activate a group of these receptors, and it's the pattern of activity across the receptors that's going to be perceived as a smell. So we have these little dangling cilia, and the dangling cilia are dangling in this mucosa, a mucus. So inside our nose we have this kind of liquid mucus that allows the chemicals, for example, to dissolve so that they can be picked up by these cilia. So these little receptor cells are embedded here into the top of our nose, and they pass through the section of our skull, which you can see has many different pores to allow them to pass through. And it's there that they go on to synapse on these cells in the olfactory bulb, starting with these ones we'll talk about called the glomeruli, or the glomerulus is a single one. So these olfactory glomeruli receive inputs from multiple receptors in the nasal passage, and any one glomerulus receives information from a certain type of receptor. So for instance, these purple receptors are only going to pick up purple odorants in the nose, and they're all going to send their information here to this purple glomerulus, which is going to send its signal up to these mitral cells that send them into the brain. So there seems to be some sort of topography, some chemotopic representation or chemotopic organization, that different types of smells are separated in the olfactory bulb here. But we don't know very much about how that space looks like, how the different smells are organized in our mental life. And there's a lot of work to be done here. But what we do know is, for instance, that across the two glomeruli, there's symmetry in this organization. So it seems to be somewhat of a consistent pattern. And so a certain odor would activate maybe purple and blue more than green and yellow, and it would then activate some of these neurons and not others. So there's particular patterns of activity in the olfactory bulbs that could predict which odor we're smelling. And we know that certain species are going to be different, but similar species are going to have a similar pattern. And within a species, there's similarity. So there's some organization here that we're learning about. Here's the olfactory pathways that I mentioned. And so you can see that the information comes into the olfactory bulb through the cribriform plate. Then it passes the information into the piriform cortex here, where the amygdala is, which we already mentioned does not first pass through the thalamus and then to the thalamus and then up here to the orbital frontal cortex, where we have our kind of primary, not our primary, but our olfactory area of our brain. But we think of this piriform cortex and the limbic system and the temporal lobe as being the olfactory one, primary olfactory area. So as we said, this olfactory pathways project to the limbic system where they mediate our emotional response to odors and our memories for odors. So we can have a certain odor, say of money, bring up all these memories of particular events or feelings. Then information goes to the thalamus and subsequently is sent on to the orbital frontal cortex, and this is where we likely can consciously perceive an odor. When you start to smell something and you're aware of it, it's because you start to get activity in this part of your brain, in the orbital frontal cortex. But we don't necessarily know, once we're in the orbital frontal cortex or the amygdala or the piriform cortex, what the receptive fields of the neurons are. And what I mean by that is what particular groups of smells or locations of smells make these neurons fire as opposed to other smells or locations. So we're still learning a lot about the olfactory system in this way. One thing to bring up is the idea of pheromones. So pheromones are smells in animals that they give off, that influence the physiology and behavior of their conspecifics, other animals of their species. So these smells are shown to regulate interaction socially. For instance, sex and aggression in hamsters can be modified by putting different types of smells. Or you can also take out the olfactory bulb of a hamster and they lose some of these smells. For instance, if you have an unfamiliar hamster in the cage with them and you put pheromones from a man or pheromones from a lady hamster, the hamster will interact with it differently, and that's mediated by its olfactory bulb. In humans, there's much less strong evidence that these pheromones exist and that they influence our behaviors, but there's some indication, for instance, that based off the smell of someone's breath or their sweat, we can identify their gender. But we don't really have strong evidence that the odors we give off are sexually attractive in any way. You wouldn't necessarily say that your boyfriend or girlfriend's underarm smell is an attractive smell. But there's lots of work to be done here, and it's a big industry, of course, as everyone would love to have a particular cologne or perfume they could put on that would make those around them feel attracted to them. But it's likely that we don't have this affecting our behavior as much as animals do. So I'll start a new video to talk about the taste system of the brain, the gustatory system. So see you in the next video.